I have the pleasure of introducing the first speaker, right? If I get it right, that's Dr. Richard Koup, who's the acting director of the Vaccine Research Center at the National Institute of Allergy and Infectious Diseases at NIH. And he's an esteemed immunologist and vaccine researcher, and he'll talk to us about the revolution of mRNA vaccines and implications for an HIV vaccine. Please feel free to enter your questions into Slido during the talk. We'll have about 10 minutes for moderated discussion afterwards.
Thank you, Elaine, and thank you, Paul and Jerry, for the invitation to come speak to you today. It's a real honor to present in this course dedicated to my dear friend, Scott Hammer. He was a man who was small in stature, but a real giant in the field, and he will be truly missed. So I work for the US government, so I have no financial interests to disclose. Hopefully, after you hear my lecture, you'll be able to describe the current status of HIV vaccine development and articulate how the application of mRNA technology may actually speed the testing of new and existing HIV concepts. What I'm going to talk to you about today is really a tale of two pandemic vaccine efforts. Obviously, the HIV pandemic, which has been going on for over 40 years, 80 million individuals infected, 36 million people have died, and for which, despite years and years of research, we still don't have an effective vaccine. And compare that to the SARS-CoV-2 pandemic that's been going on for about 2.5 years now. 522 million approximately infected, 6.2 million died, and we already have multiple successful vaccines. So the questions then become, why is there this difference? Is it all related to mRNA technology? And will mRNA vaccines revolutionize the HIV field? And I'm just going to go ahead and answer those questions right up front. And then hopefully during the lecture, you'll see why I answered these questions the way I did. So why the difference in the vaccine development for these two agents? Well, HIV and SARS-CoV-2 are inherently different viruses with vastly different susceptibility to pre-existing or vaccine-induced immunity. So we know that vaccines against SARS-CoV-2 protect against symptomatic disease and severe infection, but they don't block infection. We all know of many individuals, probably many in this room, fully vaccinated, and yet you've gotten infected with SARS-CoV-2. But luckily, the vaccines work so well that hopefully you had a very mild disease. That isn't going to work for HIV. If you get infected with HIV, you're infected with HIV. And so we really need a different type of immunity, a much greater barrier to infection for an HIV vaccine. Is the rapid development of COVID vaccines all related to mRNA? Well, no, not completely, but certainly mRNAs are much faster to develop and deploy. And will mRNA vaccines revolutionize the HIV field? Maybe not revolutionize, but they'll certainly speed the process. Okay, if we look at the development of HIV vaccines over the last 40 years, what you can see is that HIV was first isolated in 1983. It took 4 years before the first phase 1 trials of a vaccine were started. And it wasn't for another 20 years before efficacy trials began. And now we're out almost 40 years and we still don't have a successful vaccine. Now compare that to just the last 2 years where we had the COVID pandemic. And we detected the emergence of SARS-CoV-2 at the end of 2019. Within a month, we had the sequence, and then almost immediately, the US government started supporting phase 1 clinical trials, and phase 3s started just a few months later. And we had within a year, just under a year, we actually had emergency use authorization for 2 different vaccines, and then full licensure in about a year and a half. So it's been a really whirlwind type of vaccine development compared to the HIV process. So what I'll talk about today is 4 topics: lessons learned in HIV vaccine development, current approaches in HIV vaccine development, SARS-CoV-2 vaccine development, and the role of mRNA technology, and then how will mRNA technology be applied to COVID vaccine, to HIV vaccines. Sorry. So back to this slide, and I'm just going to go through the actual efficacy trials and what we learned from these, and this will be very quick. So the first vaccines were just recombinant GP120, the envelope of HIV, and what we learned is that they induced antibodies, but those antibodies did not neutralize. So antibodies that bind HIV envelope but don't neutralize, don't protect. We then had a recombinant adenovirus vaccine trial that didn't include envelope. It was GAG, POL, and NEF, and the idea there was to induce CD8 T cells and see if those could then control infection once people got infected. And in fact, we did induce CD8 T cells, but we did not slow down the disease. So CD8 T cells alone are not sufficient for protection. You're all aware of the RV144 TIE trial, which was an ALVAC, followed by a boost with GP120. And it showed about 31% efficacy. And so the thing was that this vaccine did not induce neutralizing antibodies, but it did induce binding antibodies that mediated ADCC and other things. And the thought was that maybe the non-neutralizing antibodies were protective. Unfortunately, when they tried to repeat this trial, they were unable to repeat those results. So it doesn't look like these non-neutralizing antibodies are protecting. And finally, there have been a couple of adenovirus trials, another AD5 and an AD26 trial, which included envelope, and those failed to protect. So throughout this, what we have learned is that HIV vaccines so far have failed to induce neutralizing antibodies and have failed to protect. So the field in general is moving towards the fact that efforts should be directed towards developing immunogens that stimulate neutralizing antibodies. But that's a difficult thing to do. And that's because HIV has these multiple variable loops. The envelope is very highly glycosylated, and it's hard to make a good immune response against glycogen molecules. You really want to attack the protein molecules. These glycogens tend to shield the neutralizing antibody domains, and there are multiple clades of HIV, all of which are different enough that it actually causes problems in being able to get broad coverage. So what are the current approaches in HIV vaccine development? As I said, we're trying to induce neutralizing antibodies now, and what we're really relying on is our knowledge of the structure of the HIV envelope. This is called structure-based vaccine design. So I list here a number of publications where the structure of the HIV envelope has been determined with and without glycosylations, and what's been done to actually stabilize that envelope trimer into a very stable format. These are mostly called SOSIP trimers. The SOSIP stands for the modifications that have been made in order to stabilize that trimer in its pre-fusion complex. Nice study from Rohir Saunders and John Moore here in New York City at Cornell, showed that if you take envelope trimers that are not well structured. So they're sort of, you can see in these cryo-EMs, they're very floppy. The different trimers sort of hang out there in different planes. And you immunize rabbits Or you take these SOSIP trimers, where you can see this very ordered structure of them, and you immunize rabbits. The only place where you get neutralizing antibodies is with these very structured or very, very tight trimer structures. Unfortunately, these antibodies, these neutralizing antibodies, are very type specific. So they will only neutralize the vaccine or the virus strain that was included in the vaccine. They won't neutralize all sorts of other vaccine or all sorts of other virus strains. So these stabilized envelope trimers are very important, but they're insufficient to stimulate broadly protective neutralizing antibodies. The other good thing about knowing the atomic structure of the HIV envelope is we can now identify where broadly neutralizing antibodies are actually binding to the envelope. And once you know that, you can then use that information to develop immunogens to try and get the immune response to target that specific epitope. So if you look there are 5 different regions of the envelope that are really targeted by broadly neutralizing antibodies. The CD4 binding site, I'll spend a lot of time talking about that. There's the V3 or N332 glycan supersite, the V3 loop you've probably heard about. There's a V1, V2 cap on the envelope, and that's another target. You have the trimer interface domain and the MPER, or the membrane proximal external region of GP41. This just shows some of those broadly neutralizing antibodies that have been isolated. So people who have been infected for many years, about 10, 20% of individuals develop these broadly neutralizing antibodies. Once you isolate them, you can then test them against a panel of 208 different viruses of all different clades and ask how well do they neutralize. And on these graphs, I've broken it down into CD4 binding site antibodies, those that target the V3 glycan, the V1, V2, or the MPER region. And each dot is one of those 208 viruses, and the further down on the graph, the more potent that antibody is against that virus. What you can see is the CD4 binding site antibodies are not the most potent, but the nice thing is these are the percent of those 208 viruses that are resistant. Some of these capture 96, 98% of the strains of the virus out there. So these are very broad. Some of them that are much more potent, like the V1, V2, miss a quarter to a half of the viruses. So they're more potent, but they miss half the viruses. If we look just at these CD4 binding site antibodies, the nice thing about these, this shows 11 different CD4 binding site antibodies and how they bind to the GP120. And if you overlay the crystal structures, they're almost exactly the same. So the CD4 binding site antibodies, they're really a class of antibodies called VRC01-like antibodies. And they contain the same immunoglobulin genes. It's a very reproducible response. So the question is, can we actually stimulate this type of antibody with a vaccine? And a lot of effort is now going into that. We know in individuals who are infected with HIV how these VRC01-class antibodies actually develop. So what happens is you have an envelope, it engages naive B cells, which have immunoglobulin on their surface, and starts to stimulate a naive B cell to become a memory B cell and produce immunoglobulin. Over time, that antibody response will change and will mature with somatic hypermutation until you get one of these broadly neutralizing antibodies. These very early ones are not very broad. It takes a lot of somatic hypermutation in order to get very broad neutralization. How does that happen? Well, how that happens is every time you develop an antibody against the virus, the envelope of that virus changes. After it changes, the antibody changes in order to now attack the new envelope, which changes, you get a new antibody response, et cetera, et cetera. So you could look at this and say, well, if you want this blue type of broadly neutralizing antibody from a vaccine, what you should just do is start with this blue antigen. And then you would just drive things in that direction. Unfortunately, what we know is that most of these envelopes that bind to a broadly neutralizing antibody don't actually engage the naive precursor. So what we've had to do is now go back and try and develop specific immunogens which will engage these naive B cell precursors. Several groups are working on this. I'm going to concentrate on one of them, which is Bill Schieff's group out of the Scripps Research Institute, where he's developed this immunogen that mimics the CD4 binding site. So you just take sort of what the CD4 binding site looks like, and you pull that out and make what he calls EOD-GT8. What he then did is he took that EOD-GT8 that mimics the CD4 binding site, and he put it on a 60-mer nanoparticle. So you now have 60 of these CD4 binding sites decorating a single particle. That's gone through a phase 1 human clinical trial. They've looked at whether that immunogen is able to engage the naive B cell precursors, get them to expand and start maturing. And the answer is yes. The results haven't been published, but they've been presented at meetings. So what we know is that this works. And what I need to tell you is, while I'm concentrating on Bill Schieff's work, there are other groups that are working on similar types of naive B cell precursor stimulating immunogens. One by Rohir Sanders and John Moore. John Moore is a local New York colleague, and so I can't give this lecture without a shout out to John Moore and his group. So what has happened so far is we have this EOD-GT8, which we know initiates this process, but now what we need to do is what we call shaping and polishing. So we need further immunogens, and this will probably be in the form of these envelope trimers, to shape, polish, and ultimately drive towards a broadly neutralizing CD4 binding site antibody. The problem with this process is that each one of these is a different protein, and each protein requires 1 to 2 years of manufacturing process development before you can even get to phase 1 testing. So this pathway is going to take years to decades. So what we have learned so far for HIV vaccine development is that structural details of the HIV envelope are important. They've been crucial for designing the next generation of immunogens that can stimulate neutralizing antibodies to HIV. The native envelope trimers as immunogens are unlikely to stimulate more than limited autologous neutralizing antibodies, and we're going to need a complex series of immunogens in order to get where we want to, broad neutralization. And unless we can develop or can accelerate this development process, it's going to be decades probably before we have an HIV vaccine. So let's move now to SARS-CoV-2 vaccine development. So much like what I told you about structure-based vaccine design for HIV, structure-based vaccine design was crucial for SARS-CoV-2 vaccine development. So what we knew very early on was the pre-fusion spike structure of SARS-CoV-2. And the reason we knew that is because of years of previous work on SARS-CoV-1 and MERS-CoV. So these were two very similar viruses that people at the Vaccine Research Center and elsewhere had been working on, and figured out that really all you needed to do in order to stabilize that spike protein is put 2 prolines up at the top of the spike protein. During the fusion process, part of the protein flips up to the top. Prolines will stop that from happening. So the addition of 2 prolines kept it in its pre-fusion format. And as I said, it was prior work on MERS and SARS-CoV-1 that allowed us to know this. And so you've all heard of the S2P protein. Well, that's what the S2P protein is. It's two prolines into the spike protein to stabilize it. And because of this, once the sequence was known, 5 days later, they could start producing product and start manufacturing and doing preclinical studies. Within 2 months, this is the Moderna timeline, they had phase 1s going, 140 days, and they had phase 2. Just over 6 months, they started the phase 3, and in under a year, they had emergency use authorization with efficacy. So an incredibly fast timeline using mRNA technology. So the US government actually supported 6 different vaccine products, which are listed here. So what you'll see is that 2 of them, Moderna and BioNTech Pfizer, were mRNA, 2 were adenoviral vectors, and 2 were recombinant protein. Almost all of them used that S2P mutation, and the mRNA vaccines were the fastest because they were the easiest to produce and get out into clinical trials. The adenovirus vectors were right behind. The only reason the AstraZeneca vaccine hasn't gotten EUA in the US is because it got EUA in Europe, and they decided not to apply here in the US. And then the protein vaccines have taken much longer. And as you're aware, for the Novavax, just last week, they had the VRPAC meeting, and it was a unanimous vote that Novavax, which is a protein vaccine, should get EUA approval. We still have to hear from the FDA, but that will certainly be coming in the next day or so. I just want to show you that these mRNA vaccines, not that I have to tell you this, but they're very efficacious. You've seen in the literature these curves, efficacy curves. Even after a single dose, you can see that you're getting protection very early on. So 94% protection against symptomatic PCR confirmed COVID. Both of these curves were developed during the initial wave of COVID. This doesn't include protection against Delta, Omicron, et cetera. And as a result of that, here in the US, you can see the use of mRNA vaccines versus the adenovirus vaccines. I could show you a similar graph for Europe, and it would be similar, except there would be a smattering of other vaccines in there. So mRNA immunization, we know it induces antibody, CD8 T cells, CD4 T cells that are Th1 or T follicular helper like. What you don't want from these vaccines is a Th2 response, and it doesn't induce a Th2 response. COVID-19 data indicate from the millions and millions of people that have been vaccinated that it's quite safe. We do know about the myocarditis and a few other small problems, but very safe profile. The stability and supply chain is improving. These have to be kept at -80, but that's something that's being worked on. There is some improvement in codon selection, secondary mRNA structure, et cetera. One thing that mRNA vaccines do, once they're taken up, so first you have to get them taken up. So you use lipid nanoparticles to get them taken up, and different lipid nanoparticles will lead to different amounts of the mRNA being taken up. Then you want them to induce protein production without this inflammatory response, which can result. And so using pseudouracil and some other techniques, you can decrease that inflammatory response. The important thing with all these improvements, mRNA is not magic. It doesn't in some way induce a broadly neutralizing response. You still need the right antigen design, and I think we have that at this point. The other thing is that it's very easy to produce these. Instead of 2,000-liter bioreactors, you can do these in 40 ml flasks, and it's very, very conducive to low and middle income country manufacturing. What type of immunity is correlated with the protection that I showed you? Well, this was published in November of last year, and it's neutralizing antibodies. So if you look at the peak immunogenicity after the Moderna mRNA vaccines, the higher your neutralizing antibody titer, the greater and greater your vaccine efficacy. So for SARS-CoV-2, what we've learned at this point is that prior work on SARS spike structure and the ease of protein stabilization were very important. Use of mRNA and viral vectors to express the spike led to more rapid development. And the rapid mobilization of the US government to support pharma and biotech was also crucial. High efficacy against symptomatic COVID was achieved by multiple different vaccine platforms. Induction of neutralizing antibodies by vaccines correlate with the protection against COVID, but you all know that the vaccine protection was incomplete and waned in the face of the virus starting to escape that immunity and new waves of virus variants coming up. So finally, in the last 5 minutes, how will this mRNA technology be applied to HIV? Well, on the same week that Janssen announced that its Ad26 vaccines were not successful, Moderna announced that it was getting into the HIV vaccine field. And not only Moderna, but also Pfizer BioNTech is also getting involved. How are they getting involved? Well, for Moderna, I showed you the fact that what we need is to develop these new immunogens for shaping and polishing. And in fact, Moderna has agreed to start making these different vaccine components for the Bill Schief program of HIV vaccine development. And changing to mRNA will really shorten that design cycle. For every protein that needed to be produced, you produce these out of cellular cell culture. So along with the vaccine that gets produced or the protein that gets produced, you have all the cellular components. You have whatever viruses were contained within that cell line. You need to go through a process to show that you've got very pure protein, and in the process of purifying that protein, that you haven't changed the structure. Novavax, the reason it's taken them so long is when they first started out, they were only getting 30% purity of their protein. Then when it got up to about 60% purity, that's when they were able to do the clinical trials. And ultimately now they're at much, much higher purity. But for proteins, it takes a long time. So in addition to Moderna and Pfizer BioNTech, there are lots of other companies and groups that are getting involved. Some nonprofits, such as Greenlight Biosciences, Acagera. Afrigens is a group that's trying to develop this technology specifically for the continent of Africa. And government and academic centers like the Vaccine Research Center, the Duke Vaccine Institute are getting and acquiring mRNA technology. So the important thing, however, is that don't think that Big Pharma is throwing a lot of money into this. They're still looking for the US government and others to fund it, but they're allowing their technology to be applied. So what can mRNA technology do for HIV vaccine development? Well, shorten timelines for production to clinical testing of vaccine concepts, eliminate time-consuming and costly steps associated with protein vaccine manufacturing, and decrease cost of production. This will shorten the overall design cycle time, which is the time from a vaccine concept to an actual product that goes into the arm of an individual. What it can't do is induce some sort of magical sterilizing immunity. The SARS-CoV-2 mRNA vaccines protect against symptomatic and severe disease, but they don't provide sterilizing immunity. And we've seen that these progressive waves of infection within vaccinated populations clearly demonstrate that the current mRNA vaccines do not provide robust protection against infection, something that's going to be needed for HIV vaccines. So just to finally re-answer the questions that I posed, why the difference between HIV and COVID vaccine development? Well, SARS-CoV-2 and HIV are very different. A vaccine that provides neutralizing antibodies and protection against some infection, but not all infection, is good for SARS-CoV-2, not going to work for HIV. Is the rapid development of COVID vaccine all related to mRNA technology? No, but mRNA will help. And will mRNA vaccines revolutionize the HIV vaccine? Maybe not revolutionize, but certainly help speed the process. And boy, we need new concepts and faster testing of these HIV vaccine concepts. And with that, I will just leave this slide up there. There are too many people to list individually, but just say that this has been a real incredible group effort, and I'm happy to take any questions.
Yeah. So great. So I'm going to encourage everybody, if you have questions, to put them into the Q&A. We have a couple that I'll start with, and the first question is whether the availability of the Novavax vaccine is helpful. Are there any advantages? How do you see that?
Yeah, that's a very interesting question. I mean, Novavax is a protein. The initial thought was that there's a group of people out there who didn't want to take the vaccines because it's mRNA, it's new, there's not that much known about it, and that they would be interested in getting a protein vaccine. I think that's probably a very small part of the population. The other thing that still needs to be tested, we know from mRNA and the Janssen Ad26 vaccine that when you boost, you get very good immunity, but then that drops off fairly quickly. So there's the question of whether the mRNA vaccines are just not providing enough durable immunity, and maybe boosting with a protein vaccine will give more durable immunity. We don't know the answer to that yet, but certainly something that will be tested.
Great. So another question is, do you think the mRNA technology will allow for better flu vaccines?
Yes, very good question. So as you know, for the flu vaccines, every year they have to figure out in the Southern Hemisphere what may be coming next year. You then need to start production of what you think is going to be coming. Some of that's produced in cell culture, some of it's produced in eggs. You've got all of the manufacturing, all of the purification, and it takes a good 6 months to do that. So you're really looking 6 months ahead of time to try and predict what you need to manufacture. If mRNA can shorten that to 3 months, it allows vaccine that we're that we're going to be administering in the fall a little better to what actually ends up circulating. As you know, sometimes we get it wrong. We have the wrong strains in the vaccine.
Great. So another question is, as we develop and implement better and better PrEP agents, antiretroviral agents, maybe being, how do you actually test the efficacy of new these new vaccines?
Yeah, that's an excellent question. I mean, we have so many prevention strategies, so many of which work, that it really increases the size and complexity of any efficacy trial that we need to do. So I'm not going to claim to be a clinical trialist. There are a lot more people in this audience who know this better than I do. But it's not going to be easy to test efficacy of HIV vaccines.
They're even having difficulty testing new PrEP agents, given
yeah.
currently available agents. Yeah.
Right.
So we have another question. Will the development of vaccines using mRNA technology go after the different targets within one or a series of vaccines, CD4, MPR?
I'm sorry.
Yes, so I'll read it again. Will the development of vaccines using mRNA technology go after the different targets?
Very, very good. Yes.
Yeah.
So as I say, I concentrated on the CD4 binding site antibody lineage that Bill Schief and his group are attacking. The John Moore immunogens are actually targeting not only the CD4 binding site, but also the cap, the V1, V2 cap, and potentially other sites on the envelope trimer. So as mRNA technology comes in, yes, those other targets will be part of that development.
Okay. And then do you think the mRNA technology will allow us to one day develop a successful vaccine against the common cold?
Well, I think coronaviruses are one of the causes of the common cold. And yeah, I think coronaviruses aren't the only thing. There are lots of other viruses, but the question becomes, how many different vaccines can you put into a single, or how many different viruses specificities can you put into a single mRNA vaccine? And the nice thing is that you can package a lot into mRNA. And so, you could have a vaccine that covered a few different human coronaviruses, a few different adenoviruses that cause common colds. So yeah, the world is your oyster. Who knows what we can get to?
Okay, so speaking of flu vaccines, and it's fine for you to say it's not your turf, will the 2022-23 season flu shots include protection for the newer strains of COVID?
So it'll be separate. I mean, what we're trying to do is have the new COVID vaccine to have the rollout of a COVID vaccine boost for the fall. There is a VRPAC meeting on June 28th, which will decide what the component of that will be. It may include Omicron in addition to the original strain. And then that would be rolled out at the same time as the flu. It wouldn't be combined with the flu vaccine, but you could go to your doctor, you could go to your drugstore and get both vaccines at the same time. That's the hope.
Maybe that'll increase the number of people who get both. And let's do one final question. What would it take for a COVID vaccine to prevent transmission? That's
a very good question. It's sort of the next wave. We're almost starting from the very beginning in development of this. So what we know is that it's very easy to protect with the current vaccines against lower respiratory tract infection, but we're not really protecting very well against infection in the nose or the amount of virus that's shed. So what we're trying to do now is develop vaccines which are given intranasally and stimulate mucosal immunity such that hopefully will block a lot of the infection within the nose. That will block transmission to other individuals. But we're just starting on that journey right now.
Well, on that fascinating note, I'll thank you for a fantastic talk.
Thank you.
And for answering questions. Thanks. And So we'll take a brief break and I'll ask you to go back into Slido and to answer 2 more questions here. How many individuals with HIV are presently under your direct care? Music. Okay, let's see what we have. Fantastic. So about 1/5 of people are not providing direct care, but let's see, you're still going. Okay. So we clearly have quite a few clinicians and some of you with very large practices. So fantastic. And we have one more question. How many in-person CME activities have you attended in the past 2 years? Know my answer to that, I told you earlier. Okay, let's see. So welcome particularly to those of you who are here, your first CME, live person CME, and wow to those of you who've been doing this over the last couple of years. That's fantastic.